in his famous book, Blood, Pure and Eloquent, Dr. Wintrobe described blood as a living substance, the very essence of life. Blood is highly active and is indeed thicker than water, because suspended within this constantly circulating fluid are billions of cells. To maintain constant blood volume and function, 500 billion cells are created every day. If we examine one drop of healthy peripheral blood under a microscope, we can distinguish several different cell types, the erythrocytes, the thrombocytes, the leukocytes, each of which has a specific life support function to perform. But where do all these cells come from? Every cell in your peripheral blood is derived from the hematopoietic stem cell, the mother cell that resides deep in the marrow in your bones. Thus, the stem cell is pluripotent, containing within her walls the potential to differentiate into any of these other highly specialized cells. The thrombocytes, which contribute to hemostasis, healing injuries, and therefore maintaining blood volume. The erythrocytes, which transport oxygen and glucose, amongst other substances. The myeloid leukocytes. The granulocytes, basophils, neutrophils, and eosinophils, and also macrophages. The myeloid leukocytes are involved with innate immunity, which protects us against infectious pathogens. And finally, the lymphoid leukocytes, the B and T lymphocytes. These form the basis for the acquired immunity that you build up through environmental exposure. So the question we might ask is how? How does one seemingly large and rudimentary cell evolve into all these very different cells? Well, the process of hematopoiesis is triggered by changes in gene expression as the stem cell matures. These cause the immature cells to differentiate through one of 4 pathways, indicated here on the chart. As cells reproduce, they evolve, changing their shape until they reach their mature form and are released from the bone marrow and into the circulating blood. This is a complex, large-scale production process, during which multiple precursor cells are formed as each lineage develops from the immature to the mature state. Now, as with any production process, there is always a possibility of errors. In this case, mutations in the DNA code that arise during the replication process. Our hematological quality control is apoptosis, the mechanism by which mutant cells are identified and destroyed. Now, occasionally, a mutant cell slips through the net. For example, in the case of leukemia, where an immature leukocyte mutates in such a way that it proliferates. Excuse me a moment. Sorry. What is it, Ali? Well, you can handle this. Get Martin to clerk him in, take bloods and an aspiration. Look, I've got to go back in. Well, what's his white blood cell count? How high? 144,000. Oh, Ali, I'll be right there. It's called acute lymphoblastic leukemia. Well, yes, it is very serious, but we do have treatment options. So, we'll start you on chemotherapy, and if the disease responds as expected, we should get you into remission in about 6 weeks, which means the malignant white blood cell count should be near 0. Well, unfortunately, in this type of leukemia, remission doesn't tend to last very long. It's Philadelphia positive, which means it's particularly aggressive. You do have the advantage, though, of being young, fit and healthy, apart from the leukemia, of course. And you are in good hands, but you need to prepare yourself for a long and difficult road. Do you have a brother or sister? One of the treatment options is a stem cell transplant. To get you into permanent remission, you'll need more chemotherapy, this time myeloablative chemotherapy to wipe out the diseased cells lurking in the bone marrow. Then, that will be followed by radiotherapy to make sure there are no dormant leukemic cells in the brainstem. Now, these treatments will deplete your healthy cells as well as the malignant ones, which will leave you immunocompromised. And that's why we need to do a transplant, to reboot your own hematopoietic system with donated cells. Of course, there are some risks associated with transplant, but on balance, it is our preferred course of action, if we can find a donor. Negative complications of a stem cell transplant include infection, graft versus host disease, and graft failure. Each of these is potentially fatal. However, for diseases with poor prognostic outcomes, such as leukemia, a stem cell transplant represents the best chance of long-term survival and cure. Therefore, the risks of a stem cell transplant are outweighed by those of the disease itself. A transplant is therefore the optimal therapy for patients who are strong enough to withstand the consolidation therapy and who have a matched donor. A matched donor is someone who is genetically compatible with the recipient. A good level of histocompatibility reduces the risk of graft versus host disease, which is when the donated cells recognize the patient as foreign and attack him. to ascertain whether someone is a compatible donor, we look at human leukocyte antigens, or HLA. Antigens are proteins that appear on the cell surface, and this specific set of HLA antigens, located on chromosome 6, determines whether or not two people's immunological responses are compatible. There are two classes of HLA antigens. In Class 1 are A, B and C. Class 2 are D, DQ and DP. If two people match at each of these 6 loci, we call it a 12 out of 12 match. However, each HLA antigen also expresses information through its various alleles, which is called allelic polymorphism. Currently, we have identified 1,884 alleles for locus A, 2,490 for locus B, 1,384 for locus C, 1,194 for locus DR, and 165 for locus DQ. Therefore, the chance of finding a possible match is 10 to the power of 23. This is a serious obstacle to transplant, and the reason we always start with the patient's siblings when looking for a donor. There is a 1 in 4 chance of a sibling being a match. For patients with no matching siblings, we look through the bone marrow transplant registries, which keep records of the HLA data for potential donors. Worldwide, over 20 million people have joined a stem cell registry. The chance of finding a match with a voluntary donor is 50%.
Comes to your office or home and gives you injections to stimulate your blood stem cells over 3 days. They'll sit with you for an hour each day to check if you're comfortable. Over those 3 days, you'll probably notice your bones aching. That's as the stem cells emerge from where they're located within your bone marrow, flooding your bloodstream. They normally emerge if you have a major trauma and your body needs repair, so it's quite natural. On the 4th day, you get a train to London with a companion. You'll have a 4th injection in the hospital, then go to your hotel for the night.
The donor's blood is being drawn out of the body from one arm and into this machine. Down here is the centrifuge. There's a belt and a spinning action. So the blood that's coming into the machine is spun in order to separate out the stem cells. These stem cells are collected in a closed system bag. In this tubing here is a blood warmer, so the donor doesn't get chilled when the blood goes back into her other arm. These are the bags. One is saline, one is an anticoagulant, and one is calcium. At any given time, about 280 millilitres of her blood is in the machine, coming out of one arm, through the machine, and back into the other arm.